This video supports application E Forensic Genetics in the Saunders textbook. First, let me say there's no substitute for reading the actual chapter. It contains a wealth of knowledge and stories which one should be able to relate to pretty easily. So that's my first suggestion. Failing that, you can then listen to this video and understand some of the areas that you may not understand fully. One should be familiar with the fact that one's own DNA can be used to identify on a pretty unique basis an individual. The only exception is identical twins, where the egg and the sperm have split to give rise to two or more offspring. In all other cases known so far, the DNA of every conception is unique. This identification relies on differences in the arrangement or basis of our DNA. Various names are used in the literature when referring to this technology. The most common these days is DNA profiling, but other papers or textbooks may use the terms DNA fingerprinting, an antiquated term, forensic genetics, and even forensic genetic analysis. Since the first known utilization of these techniques in 1986 by Alec Jeffreys of the University of Leicester, many refinements have been made, and currently we have methods that allow easy identification using carefully documented and selected markers along the DNA. Markers are simply landmarks that can be used to identify one type of DNA from another's, and the application of mathematics to quantify these values. Students are expected to perform just very simple arithmetic in order to deduce the probabilities. The authors have divided the chapter into three parts. The first part deals with crime scene DNA. The middle part looks at paternity testing and examination of DNA for individual identification of remains. And the final part of the chapter talks about direct-to-consumer kits available from companies such as 23andMe to identify the risk of either developing a disease or discovering one's ancestry. Beginning with the first of these three applications, let's look at forensic genetic analysis for crime scene data. The Federal Bureau of Investigation has been key in working with scientists to develop more robust and reliable kits distributed outside the agency and available within the agency for use in identifying individuals. The Federal Bureau of Investigations has decided to examine two types of variation found in human populations. The first type are short tandem repeats, STRs. These are generally less than 10 base pair repeats that occur at various locations throughout the human genome. The alternative candidate of polymorphism, known as variable number of tandem repeats, VNTRs, also occur at a high frequency of variation within the human genome. These type of repeats are generally longer than 10 base pairs in length. Of the two, the FBI decided to go with STRs. STRs normally are about 4 base pair tandem repeats. So in this example here, we have ACC as the call repeat that's been repeated 10 times in this example. The process entails the application of PCR amplification on DNA from suspects and crime scenes, and once the DNA has been amplified, the DNA is then separated on a gel according to size. Barring mutations, which are very rare, each one of us has 2 chromosomes. As with other genes, we can have 2 copies that differ, or we can have 2 copies of this gene that are the same. So within the population, different alleles exist. For instance, the population may have 10 repeats. They may have 9 repeats, 8 repeats, 6 repeats, 5, 4, etc. So using that as an allele, we can then predict through investigation what size alleles somebody could inherit, or compare the alleles found in the sample at the crime scene to suspects. As the FBI's technologies improved, in 1990, the FBI began to release these kits called CODIS. And CODIS is an acronym for Combined DNA Index System. These kits contained enough material to test 13 different loci simultaneously using similar conditions of PCR on any DNA sample. Additionally, scientists also calculated the frequency and number of alleles found for most human populations. As an extension of this work, Congress in 1994 passed legislation called the DNA Identification Act, which set in place the rules to which the FBI adhered and came up with a national DNA indexing system, NDIS, using CODIS markers. This allowed the exchange of information between the FBI and the states, as well as local, national, and international law enforcement agencies. These databases have continued to grow almost exponentially since, and they contain a wealth of knowledge about those participants or suspects that have been required to supply DNA samples. Even though not required to do so, the FBI scientists in 2017 added 7 more STR loci to the existing 13 to make 20 markers, which are now spread out at various locations over the human genome. This addition further strengthens the durability of the data in the legal system. The output from a CODIS analysis is indicated here as an example. In the top panel, we can see that we are testing for 4 different locations on the human genome. The D stands for DNA. The number after the D stands for the chromosome on which this marker is located, the loci, and the numbers thereafter indicate that the S stands for a single location, and the numbers thereafter are the exact location, the address on the chromosome at which this loci appears. Since the vast majority of humans have 2 chromosomes, the maximum number of peaks from a single DNA sample would be 2, as seen here for CSF1PO, and also for D7S820. The minimum number we can have at any one location is 1. You could have 2 chromosomes that carry the same number of repeats, as in the case here for D8S1179. So this individual will have 2 peaks at size 12. In total, we have 13 different locations represented by all this data here. And you can tell that by looking at the labels for the markers. And also above the graphs, you have the size of the PCR fragments that are generated by the computer software. One thing to notice is that some of these numbers do overlap, 110, 110, 110, up to 350 in each case. Therefore, what would prevent the mixing of different loci in this analysis? And the answer is that in the CODIS kit, the fluorescence that's used is different colors. So this marker here, this location, and this location would use different colors so that the computer will be able to discriminate between which one belongs to which locus. When the engineers are working on developing these kits, and maybe into the future, they have to adhere to certain rules that must make it easy for the users to utilize these kits. So all these STR markers in the CODIS kit must abide by the following 4 rules. Number 1, they have to have a location that's known within the human genome, and that location and its alleles must follow Mendelian principles and are sought independently of any other markers that are present within the genome. Second, multiple alleles must exist in the population. Not only that, they must also occur with a certain frequency within two parameters. The lowest level is 1%, and the highest level should be about 25%, although occasionally different populations in the planet, for instance Eskimos, may have a slightly higher level than in most other populations. The third and fourth rules are more technical than they are genetic. The alleles must be consistently available and reliably amplified by PCR. So if that region of the chromosome is difficult to amplify, then it would not be useful in the CODIS regimen. And second, once those amplified fragments have been obtained, they should be easy to analyze through high input automated robots. And lastly, there should be no error in discriminating between different alleles. So this difference should be easily distinguishable. There should be no overlap or confusion as to size of the bands. The best way to relate to this technology is to look at a number of different examples. So that's exactly what the textbook does. It gives us some examples. So here's an example of DNA from a single individual that has been amplified up just using 3 STR markers in the CODIS regimen. One of those is the D3S1358 locus. The second one in here is the BWA locus, and the last one is FGA locus. For this one here, the first of these loci, we find that in the population of humans on the planet, the size of the alleles ranges from a 12 repeat to a 19 repeat. And the frequency, the average frequency in the population, goes from 27% for size 15 all the way down to 1.5% for 3 of these markers. Looking at the middle one, the allele frequency there ranges from 25.7% again down to 1.5%. And the size of the repeats is from 12 to 20. And finally, for the last marker, the number of repeats goes from 18 to 27, with the highest rate represented by 21% here for 22 repeats, and the lowest is here for 1.7% for 18 repeats. Before continuing with this video, please make sure you understand the logic that's used in representing this data. Put another way, every single human on the planet has these 3 loci, and each one of us will have at least one of these alleles present on one of our chromosomes. So if you have 2 chromosomes, you may have a 12 repeat from your parent, father, and you may have a 17 repeat from your mother. Or somebody else could have 2 repeats the same size from both their parents. And the frequency at which these repeats are present in the population goes from 27% all the way down to 1.5%. Once a sample of DNA is available, and these days we can do PCR on as small a sample as a single cell, the amplified PCR products are then run through a capillary gel electrophoresis. This is a simple glass tube to which a computer loads and passes the DNA fragments. At the other end of the capillary is a laser beam which excites the molecules of different colors and records the size of the fragments. And as we mentioned earlier, the output is such. The computer program will print the size of the highest peaks that it determines onto the printouts. The underlying logic comes from simple genetic analysis and the way that alleles move between one generation and the next, and the frequencies that exist in populations. What the final analysis reveals is the probability, just the probability, that a person selected at random from a certain population has that specific genotype combination. As you will see in the next example, this may not sound that robust, but once you start looking at multiple loci, multiple locations for these markers, you'll see the power of this technology. In the crime scene scenario, the suspect DNA, or the samples collected from crime scenes, are compared to the database of data collected from various agencies. The logic is very simple. If one of the anticipated bands or peaks is not present in the sample of DNA from an individual or suspect, then that person must be removed immediately from the analysis and excluded. So the principle of exclusion means that a single locus is enough to eliminate somebody as being the suspect, and they can go home. Another example illustrates this perfectly. Suppose we go back to the original 3 samples that we looked at here, and using these frequencies for suspect DNA, we continue to calculate for this individual, a suspect, that he or she has a 17-19 genotype for the first marker, a 14-14 genotype for the second marker, and a 20-25 genotype for the third marker. How do we calculate the probability that we have the suspect? And the answer is straightforward. Using Hardy-Weinberg principles that refer to the alternative types of frequency for alleles in a population, for instance, with 2 alleles, the homozygous condition would be P times P. The alternative homozygous condition would be Q times Q. And the heterozygote would be 2 times P times Q. And if you're not familiar with what is being said, that's okay, because another chapter deals with Hardy-Weinberg equilibria. But for now, just accept that the mathematics has been tested and proven multiple times. In this example, we take the frequency of the 17 allele, multiply that by the frequency of the 19 allele, times that by 2, and we end up with a number. We do the same thing for the second locus, the VWA, and we find that the 14 is present in the population at 13.4%, and because it's p squared, we use that number twice, the same number for each allele. And likewise, for the FGA, the frequency of the 20 repeat is 12.5%, and the frequency of the 25 repeat is 9.4%. And we times that by 2 because we have a heterozygote. Then we end up with this number here. The beauty is that you can now multiply the 3 numbers together to obtain the probability of somebody else having the same DNA in that population. And the answer comes out just for 3 loci to 2 in a million. So 2 people in a million would be expected to have this particular combination of alleles for these 3 loci. So you can now see the potential of small numbers to be multiplied together to give you huge numbers. Let's expand this analysis to include a crime scene where we have 5 DNA samples. 1 from the crime scene itself, 1 from the victim of the crime scene, and 3 potential suspects who may have been identified as being there. So here's the data. Here's the victim's profile, and this is the crime scene profile. The crime scene profile can help us eliminate the DNA of the victim, but in this case it doesn't seem to be contaminating the sample. And then we have to compare the 3 suspects to the crime scene. You can see immediately that suspect 1 has bands for the first locus that are not compatible. Therefore, right away, he can be eliminated or she can be eliminated. Likewise, suspect 3 has a different pattern for the first locus, and they too are asked to leave. and are Fred. Suspect 3, however, for the first locus has a good match. For the second locus, they have a match, and for the third locus, they also have a match. In this case, further testing would be necessary for further loci using the CODIS kit, and if they continue to agree, then we've caught the suspect and we can send him forward for trial. Figure E3 goes on to look at a further 4 loci in addition to the first 3, and using this we can then calculate the mathematics, and believe it or not, the numbers come out to be 3.52 in 10 to the minus 13. That means the probability of somebody else having the same genetic profile as the suspect is over a trillion, 1 in a trillion. Table E3 shows us how this calculation was obtained. It's exactly the same logic as we did for the 3 loci. Now we do it for the 7 loci. So every time you have a heterozygous condition, multiply the 2 frequencies of the alleles in the population by themselves, and then don't forget to times by 2. And every time you have a homozygous condition, take the allele frequency and multiply it by itself. Once you've done that for all 7, then these values are multiplied by themselves all the way through 7 times. This new value can then be either presented to the suspect or to the court. Part 2 of the chapter focuses on paternity testing, much like one sees on daytime TV. In this case, the technology can also be applied towards the establishment of identity in selective breeding programs, pedigree analysis, and husbandry of animals. The important factor is that once you know the mother's or one parent's genotype, and you know the genotype of the offspring, the baby, then anything that's left over inside the baby that doesn't belong to the mother, and we call that the non-maternal markers. Therefore, every single non-maternal marker must be carried by the father in order to have passed it on to the child. And we'll see that in the examples that come. Again, this is a probability calculation, and in this case we're trying to determine the likelihood that another man carries the same alleles as contributed by the father. Once more, examples shall be used to demonstrate paternity testing. Example Analysis E.4 from the chapter presents a case where we know the CODIS STRs for the mother, we know them for the child in question, and then we have 3 potential fathers whose CODIS STRs have already been calculated. And we're trying to find the probability of paternity from these results. This time a special index is used to relay information. The first is called the paternity index, the PI, and that is simply a ratio. The numerator, which goes on top of the equation, is the probability that the non-parental alleles came from the putative father. And the denominator of the ratio is the probability that the allele came from another male in the population. Once that's calculated for each of the loci, then we can combine them into something called a combined paternity index, CPI. And that's simply a multiplication of the individual PIs. And once we get a CPI of greater than 100, that indicates a 99% or even higher probability that the man is the father and there is no other. The key is what value to use in the numerator. If the father for that loci, for that allele, is heterozygous, then we use a value of 0.5. If he is homozygous for that allele at that locus, then we use a value of 1.0. and then the value for the allele frequency for that loci is taken from these tables here, the same tables that we used earlier. Working through the example with the F1 putative father, let's have a look at the data. Looking at the FGA locus, we find that the F1 father has 2 alleles. Therefore, he's heterozygous. He has an allele 20 and an allele 24. So, for that locus, FGA, allele 20 has a frequency of 0.125. Therefore, for the FGA allele that he passes through to his child, so let's have a look at that first. The child is 2023. The mother is 2324. The child must have got 23 from the mother. There's no way the child could have got 20 from the mother because she doesn't have that. So where did the 20 come from? Let's assume the 20 came from this father. So that's the allele that we're talking about. We can ignore the 24 in this case because we know that wasn't passed through to the child. So concentrating on just the 20 repeat allele in the father 1 for the FGA locus, that's the value that we look up. The same thing will happen with VWA. The baby is 1718, the mother is 1818, so the 18 must have come from the mother, the 17 must have come from the father. So in F1, all we have to do is go to this table and look up 17. And you can see here that 17 has a frequency of 0.257. Now, because the father is heterozygous, we put 0.5 as the numerator and that value of 0.257 as the denominator. And we do that calculation. You do that all the way through, and then you add up, you multiply the values by themselves, and then you see if that number is greater than 100. If it is, then the likelihood is that F1 is the father. With individual identification, the goal is to shift through possibly multiple samples and identify each one individually. And that's the case that we have with the next story. Everybody should understand that during the 9/11 attack on the World Trade Center, 2,753 people were killed when the buildings collapsed. Some bodies were recovered and identified, but unfortunately, because of the way the buildings came down, most were not. In the subsequent investigations, over 22,000 unidentified remains were collected, mostly dentals and bone samples. And the goal was to identify each one as belonging to one of those victims. So allelic DNA from personal items such as toothbrushes was compared to that from the recovered remains. The location of the loci can be helpful in filtering down further information. The most common source of genetic markers are on autosomes, the non-sex chromosomes, of which humans have 22 pairs. However, genetic markers located on the Y chromosome can be used when the victim is a male and the reference sample from the victim or DNA from the father or the son of the victim will also contain the same Y chromosomal DNA. So by following the transmittance of this particular chromosome and its markers, we can then identify through that process which remain belongs to which male. Now for females, the situation is more complex. One solution is to follow mitochondrial DNA, which is transmitted from mothers to all their children via the egg. So if you have a remain and you can identify the potential victim by looking at the mother's sample and then comparing it to the remains in terms of mitochondrial DNA, and that should identify who the offspring of the mother are. Employing these particular types of techniques, the scientists have identified 1,637 of the 2,753 victims as of the publication of the textbook in late 2016. A second story with similar application was the disappeared in Argentina. According to history, in 1976, a military coup replaced the lawfully elected Argentinian president. And in the subsequent years, the military leaders were responsible for arresting, kidnapping, and killing a large number of opponents. Two groups of concerned citizens set up individual groups, and they asked the geneticists to assist in identifying the remains as well as the offspring of these killed individuals. One of these geneticists was Mary Claire King, and she worked with both groups, and she was able to identify more than 100 illegally adopted children using mitochondrial DNA from the grandmothers, with the same assumptions we saw on the previous slide, that mitochondrial DNA is fully inherited by all the children of that mother. The final part of the chapter addresses the genealogy trend, the ancestry seeking trend, and the health risk assessment trend, which is quite popular at this time. The authors of the book have placed these under the title of direct-to-consumer DNA testing services. The technological trends towards DNA chip-based assessments has reduced the cost and increased the throughput, allowing these companies to process huge numbers of samples without too much difficulty. Instead of testing 20 loci, these kits allow hundreds of thousands of SNPs to be tested simultaneously. All that is necessary is a sample from the customer. Normally by a swab of the cheek, internal cheek, or by spitting saliva up to a certain volume into a tube and mailing that in. Now, the number of these SNPs screened by different companies varies, as does the accuracy and the interpretation of the results. Looking at genealogy relationships first, one needs to define what genealogy is. So a genealogical relationship is a genetic connection between related individuals as a result of shared ancestry. So the descendants of individuals produced through sexual reproduction will inherit, in most cases, the same alleles that were present in the parents, as we saw earlier. There is a caveat to this technology in that in order to establish identity by descent, a decent number of haplotypes have to be analyzed. What is a haplotype? It's a region, a physical region of the chromosome that carries a particular combination of these genetic markers. The descendants of individuals will tend to maintain the order and the alleles of these regions as they are passed from generation to generation. Occasionally, crossing over will tend to disrupt some of them, but those can be then identified. The regions that are studied for haplotype analysis normally are greater than 7 centimorgans. Mathematicians have told us that anything shorter than this will not be statistically appropriate to establish identity between individuals. Applying the same logic as we did with Mendelian genetics, the number of these haplotype segments that are shared and the total length of segments can indicate the degree of closeness of the relationship between individuals. That means closely related individuals will share more of these than random people or more distantly related relatives. So first degree relationships are those where we have parents and children, where they share 50% of their alleles. So if a father and mother have a child, 50% of the father's haplotype will be passed to the child, as well as 50% of the mother's haplotypes. The next order of relationships, known as second-degree relationships, that will be brother and sister, grandparents to the child, and grandchildren of parents. These share 25% of their genes. Third-degree relationships will share 12.5% of their genes. That will be uncles, aunts, and first cousins. Ancestry is not the same thing as what we just discussed with genealogy. Ancestry is basically looking at the origin of one's ancestors in a geographical setting. Scientists seek the purest relationships available to set the foundation or the baseline for a geographical region. For instance, Aborigines. So if you know that a particular group of Aborigines have been living in the same location for a period of time, their DNA is the most valuable to use as the reference genome representing that area. Once the computer software has this data, it can then reference the customer's DNA to those particular markers. Utilizing complex algorithms and calculus, the companies are able to relate the haplotypes from various regions of the customer's DNA to these reference points. And therefore, they're able to tell you what percentage of your genome belongs to which geographical region. Indeed, over the last few years, the availability of DNA from Neanderthals has further enhanced the availability to compare genomes. And now these companies can tell you what fraction of your genome is Neanderthal. This is quite amazing and fascinating at the same time, although it is still in its infancy as a fully fledged, reliable method. Finally, genetic health risk assessment has now been offered by companies such as 23andMe, who have got a FDA approval to make such analysis available to the public. These assessment estimates are nothing more than association studies. Association means that one item is generally more likely to be found with another without having 100% correlation. So if you have a landmark area on your DNA close to where you think the condition or the genes for that condition lie, and that marker is present in a significant population of those afflicted with the illness, then you have a method of measuring that assessment. Unfortunately, unless you know 100% the relationship between the genes and its alleles and the genetic condition, these assessments are nothing more than a probability analysis. Two examples are provided. The first is Alzheimer's disease, the late-onset version, abbreviated AD. Late-onset Alzheimer's disease is marked by cognitive failure, inability to remember people and names, which progressively worsens with time. The full biochemical cause of this disease is not understood yet. But we do know at this point that there is a gene called APOE, and if you have one or two copies of one of its alleles, APOE4, then there's a significant risk factor for developing Alzheimer's disease. There is a warning, however. This doesn't mean that you're guaranteed to develop Alzheimer's if you have the allele. nor does it mean people without the allele will not develop Alzheimer's disease. What we're saying is there's a probability and an association between this allele and whatever causes that particular disease. The disease itself is multifactorial, has many components to its manifestation, but so far the research suggests that if you carry one copy of the APO4 gene, you will have an elevated risk of developing Alzheimer's. If you have two copies, then the chances are that you have an even higher risk of developing Alzheimer's. Another noteworthy disease to talk about is celiac disease. Celiac disease is simply a digestive system condition where people are sensitive to the protein from grain called gluten. We now know that the immune system has a role to play in this illness. Regardless, the gene that's most often associated with the prediction of generating celiac disease is HLA-DQ. It's part of the major histocompatibility complex that controls self and non-self rejection. Two alleles, the DQ2 and the DQ8, are closely associated with celiac disease risk. And these alleles are present in about 98% of CD patients. But we don't know if these alleles are the cause of CD or just associated with it because of other factors. Because over half the population that do not have celiac disease, they may have the DQ2 and or the DQ8 allele. So we're still a work in progress, but at least we're being able to determine some kind of association. If one reads the small print supplied by these companies, they will tell you that their results are purely advisory and that the genetic component alone isn't enough to predict or associate the occurrence of a disease with alleles. Other factors such as lifestyle, behavior, related medical conditions, environmental factors may increase the risk of developing these diseases. Customers are advised to follow up with their physicians to get a deeper assessment if one is available. That concludes this chapter. Thank you so much.